I will switch to English because my next presenter is coming from Miami, and he's not literally coming. He's in Miami right now. It's morning. We are afternoon. We heard previously in the morning, and he was just woke up. Didn't even drink coffee. I believe now he drank coffee. He had breakfast and everything. So what was really interesting about his case? He's coming from University of Columbia. It's quite fresh or young company, name Evolve. Today with us is Andrew Satz. So what they've been doing is they were using AI to find out which possible candidates as a cure against COVID-19 would be. So they searched through the world database. But Andrew will be the one who will explain it better. So we will try to explain the science side of the project as also technological. So Andrew, are you with us?
Yeah, can you hear me?
Yeah, we hear you. So what's the weather like in Miami? We have a heavy rain here.
Well, Miami, there are two rules to Miami weather. If you like the weather, wait a minute, it'll change. And if you don't like the weather, wait a minute, it'll change. So right now it's beautiful outside, but I can go back in an hour and it could be pouring. So it's beautiful right now.
Okay, so I hope that we didn't take you out from the beautiful weather.
No.
Okay, so I will leave you the online stage. You have 25 minutes. Info for everybody, so you can listen to the presentation and you can start the question that we can later ask Andrew for his opinion. And maybe next time we'll bring Andrew to Croatia. Andrew, please take the floor.
Thank you. Can you see the screen okay?
Yes.
Yeah. Perfect. All right. So thanks so much for having me, Jenko. I really appreciate it. Really excited to speak with you guys today. I'm going to, as Jenko said, I'm going to be going through how our technology works, why it is that we do what we do from a business perspective. But there are some, because we're talking about, obviously, technology, data science, AI, but we're also talking about biology and medicine. So there may be some complexities in what we explain, in what I explain. So please feel free to ask questions, and I will try to answer them in real time. So our company develops artificial intelligence, and we sort of see artificial intelligence as sort of a broad scope catch-all that includes machine learning, deep learning. I generally say AI, but and we engineered it with life science data and biological knowledge. That sort of represents the bottom right of this Venn diagram. Now, our company's expertise is in machine learning and computational biology, software engineering, structural biology, and also preclinical and clinical stage development of medicine. And so while we're working on a type of drug called a biologic, which I'll explain in just a moment what that is. We're actually more specifically focused on antibodies, and I think a lot of you have probably heard about antibodies in the news, and so we focus on this. So there's a big challenge in pharmaceuticals, a very large challenge in pharmaceuticals. These are the failure rates as a drug moves from a laboratory bench, really from drug discovery, into the market, where the drugs are actually sold. And we believe that the failure to adopt a computational approach is part of the reason we see the failure rates on the screen. Less than 2% of computer scientists and data scientists are actually in the pharmaceutical and healthcare industry. And we're part of the tiny minority approaching pharmaceuticals from an AI or computational perspective. We know that data science can dramatically drive down costs and time by predicting which drugs are more likely to fail. And at Evolve, and I'll explain to you where our company name comes from in just a minute, we're using AI to transform the pace of drug discovery from nearly 15 years down to 5 years. And what we're doing is failing as much as possible in the computer, where the cost and risk is much lower than the laboratory, because any one of these failures can mean wasted time and money. And we do this by simulating computationally the process of biologics discovery. Now, before I move forward, I want to make sure that everyone understands the difference between what a biologic and a small molecule is. So a small molecule would be things like aspirin or chemotherapy. These are chemically synthesized drugs. They're often very small, something like 25 atoms. Biologics, on the other hand, are much larger, and the way that they're made is that they're not chemically synthesized. They're grown in living cells. So, for example, when your body is producing antibodies, there are cells in your body called B cells that are actually generating the antibodies. So you can see the difference in the size. Now, when you do physics-based modeling, which is the approach of basically taking two atoms, trying to figure out how far apart they are and where they sit in space, from a computational complexity standpoint, we would just, in general, I mean, this isn't exactly perfect, but I wanted to sort of simplify this. You're doing this, the computational complexity, if something has 25 atoms, like aspirin, the computational complexity is the number of atoms to the power of the number of atoms. It's the same for antibodies, but you can see from a computational complexity standpoint, this would be very, very complicated to solve. So it's very important to understand like the difference in discovery. And a lot of companies are actually applying physics-based modeling where they're using deep learning, but instead of using pixels from an image perspective, they're using voxels from a three-dimensional perspective. And so I wanted to give you this background so you'll have a little bit more frame of reference going forward. So we're at a stage today where our algorithm takes an antibody sequence and runs it through our evolutionary models. And I should point out why we chose evolution rather than binding, which is binding and affinity is if you think about, let's say, for example, the coronavirus and a drug attacking the coronavirus, binding is the drug sticking to one another, and affinity is the magnetic charge of them coming together, right? And so when we looked at the challenge of biologics in the lab, binding affinity is the first thing tested, but which is often computationally modeled in physics-based approaches. But the challenge of physics-based approaches, again, going back to the previous slide, is that you're getting an n to the 25,000th power, which again, very difficult to calculate. So, but before that actually ever occurs, before binding ever occurs, especially in immunization, the system that's being interacted with, that was generated over the millennia, is an evolutionary system. So rather than starting with affinity, which is an incredibly important byproduct of evolution, we actually stepped back and modeled evolution. And I'll explain how we did that shortly, but I wanted to give you our thinking. And so after running our evolutionary models, we perform what's a multi-site mutational algorithm. This allows us to mutate the biologics, and then we screen them using what's called in silico assays. So these are basically laboratory versions of, these are computer versions of laboratory tests. And this allows us to predict what's going to fail. And then it's at that point that we actually look at structural docking models. Now, the process allows us to generate and then perform computational versions of laboratory tests and then screen out the variants in a way that would be financially impossible in the laboratory. And we recognize that any approach, whether it's computation or lab-based, is flawed. But because we're a computational company, not antibody engineers, we're not a laboratory, we decided to work with experienced collaborators, and that's why we send our sequences. Now, I should talk a little bit about testing and training. So, oftentimes, when you're doing data science, the cost of testing is actually more computationally, the cost of training is actually more computationally expensive than the cost of testing. In the case of generating drugs, it's actually the cost of testing is significantly higher. It actually costs about $1,000 to make each drug. So think about that. Every single time you wanted to run a test on your models, every single test that you run, so the size of the n of your y, your target variable, of your test set, the n of your test set actually would cost you $1,000 for each one. So this is another challenge in the space that we're in. So let me go a little bit into the technology itself. So we use billions of public and proprietary data points across a wide range of exponentially growing data sets, including gene sequences, molecular structure, biophysical properties, and more. And we integrate all that data, and then which obviously as data scientists, you know that's a very challenging process. And we're doing very different types of data. And then we apply our algorithms and computational models really in order to extract knowledge from the data. And this process is really about accelerating and augmenting the decision making made by life scientists. So we use computational technology to reduce the time, the cost, and the high failure rate that is endemic to the pharmaceutical industry. Now, the model uses large amounts of sequence data to learn the patterns of how biologics or proteins are formed in the body against a disease or target of interest, and allows us to generate on a scale of about 1 to 2 million at a time of antibodies in a process that takes us less than 6 hours. So from a very high level, our technology works by taking an antibody sequence of interest. In this case, across the y-axis, it's a piece of a drug called Herceptin, a biologic called Herceptin. In fact, if I jump back, this is actually an image of Herceptin right here. Herceptin is a very powerful immunotherapy used against breast cancer. In fact, if it's used early enough for breast cancer, it has a 98% effective rate, which means that 98% of the patients survive 5 years. To that portion of Herceptin, we apply our evolutionary model or mutational model, and so which basically mimics the process of how the protein is formed in the body. And we use large amounts of sequence data. Our algorithm actually learns this process. So let me talk to you a little bit about the heat map you see here. So across the top, you'll see the heavy chain of Herceptin. I'm not going to get too much into the chains, but it's just a portion of Herceptin. And on the y-axis, you'll see all the possible amino acid substitutions. There are 20 common amino acids. All of us, every person, every mammal is made up of 20 common amino acids. And amino acids are represented by letters of the alphabet. Now, through a statistical process we designed, our algorithm can actually differentiate between likely and unlikely mutations, as well as the extent of a conservation of a position. For example, we've highlighted in yellow a highly conserved position. So what the algorithm is actually figuring out what is a likely mutation from a probabilistic standpoint in blue, what is unlikely from a mutational standpoint in red. And so white is considered neutral, and shades of blue are deemed mutations that may positively impact the antibody's characteristics. It's important to understand that our algorithm doesn't look at each position or change in a vacuum, but looks at each position mutation in relation to all other positions in the sequence. Now, what's interesting to note, and was one of our earliest validation points, is that the algorithm actually inferred a very important part of an antibody, which is called the CDR regions. These are actually what make a biologic bind to its target. So think about, again, a drug against the coronavirus, the CDRs are the portions that stick, right? And if I think about like my fist, my whole fist isn't coming into contact with my other fist. It's just the exposed part of it. So think about the exposed part as a CDR region. So the resulting model actually captured diversity through that region and maintained specificity. It's also important to note that the heat grid you're shown here is a first order state. So when our algorithm selects a mutation from this heat grid, for example, by substituting an arginine for a tryptophan in position 59, which we show in the blue arrows, the grid would then enter in a second order mutational state. And then we would choose the next one in a third order mutational state. And we have to use selection criteria so that the algorithm is actually smart enough to choose that. This algorithm right here represents the core of our algorithmic process, and we apply this evolutionary model to every one of our discovery projects. In fact, we don't have to do biologics. In fact, we're working with an agricultural company to create novel agricultural products. Now, one last thing I want to point out is where our name comes from. So again, we're building AI-generated models of evolution. That's why the name of our company is Evolve. But the reason that there's a Q there is that, as I said earlier, there are 20 common amino acids, and most heavy chains of most human antibodies actually start with the sequence EVQLV. There it is right there. You can see it right there, right? This is very common among human antibodies. Actually, that's where our name comes from, is the fact that it's the first of 5 amino acids of most heavy chains of most human antibodies, and that's where our name comes. So in an intersection of science and technology, we're a sci-tech company. So once we've generated, now remember in the last step, we generated 1 million antibodies. But evolution has 2 parts. Evolution has mutation and then the system, right? So if you have a mutation, does it function well in a system? So what we do to actually create that system approach is once we've generated those sequences using our evolutionary models, we characterize our antibody variants along a host of over 200 factors. For all intents and purposes, we're performing computer versions of laboratory tests, but at a scale and speed that would be incredibly challenging in a laboratory. We just generated 1 million antibodies and are running 200 computational tests. So just imagine trying to do 200 million laboratory tests. It'd be very complicated to do that. And then we actually examine the characteristics both locally and globally across the amino acid chain and analyze how our AI-generated variants compared to existing FDA-approved and phase 3 antibodies. And on the screen, you can see the FDA-approved and phase 3 antibodies in green, while our AI-generated antibodies are in pink. We try to ensure that all of our AI-generated antibodies fit within the distribution. Now, the reason we do this is it limits the likelihood of challenges or risks that could make the drugs fail downstream, either in a lab, in animal studies, or in clinical trials, where it's costly and requires a significant amount of time. And while our algorithms don't guarantee success, it's our goal to prevent issues early on in order to reduce downstream development risks. And once we complete our 200 computational assays, we actually reduced our count from 1 million antibodies down to about 10,000. But 10,000 is still too many for our collaborators to make because it costs on average about $1,000 to make each drug. Remember, our test sets are very expensive. So we cluster the remaining sequences by mutational and characteristic features in order to maximize diversity. So if our collaborators want to make 20 or 500 antibodies, we want to ensure that the antibodies we provide them are diverse and best represent the 10,000 antibodies that have made it this far through our algorithmic process. These clustering approaches are employed so that we can provide highly diverse panels for our collaborators. Now, after we've generated the sequences, screened for failure, and clustered for diversity, we perform computational structural modeling that I talked about earlier to assess the remaining candidates for binding affinity. And we're actually we predict relative binding at scale, running over 250,000 structural binding simulations at a time, which takes us about 3 days. And on the right in yellow, you can actually see the computational binding scores of the input sequence. Remember, we started with Herceptin. In blue, you can see the binding scores of some of our AI-generated variants of Herceptin, and they're binding to the target of Herceptin, which is called ERBB2. And so we're looking at that information to try to validate how well our algorithm's working. Now, today, that entire process I explained, from input of amino acid sequence to computationally generating sequences with high affinity and lower likelihood of downstream failure, takes us less than a week. And then another 5 weeks when we send our sequences to our collaborators to make the drug. So to put this in perspective, we've done this in 6 weeks. This process today takes 2 to 4 years in the laboratory. So now I want to move on to the validation of our algorithm. As I pointed out earlier, we actually work with our collaborators to to actually validate, but we're impatient data scientists. We're not going to wait those 5 weeks to get those results to try to predict how well our algorithm works. So as I explained, we began our algorithmic work with Herceptin, partially because of how long. Herceptin's been around since the early 90s, so how long and well it's been studied, but we had another motive. There's a lot of retrospective data. So in order to validate our algorithm, our team compared our AI-generated and screened Herceptin variants to a global patent database of over 30 million biologic sequences, which offered us one method of performing a retrospective analysis of patented variants to Herceptin. And these were the results. So when we did 100% matches of our AI-generated variants to patented sequences, which pass, all of these are AI-generated variants which pass the in silico screening, and we identified 343 antibodies that were AI-generated that had already been patented. This is very important because it means that our algorithm's actually generating drugs that companies are investing money into patent. And remember, Herceptin's been around a long time, so it's been studied a lot in the laboratory. So first is that first thing I should note, the patented sequence database was not part of our training data, so these were actually unbiased results. And second is that the point when we performed this validation, our algorithm that actually checked binding wasn't ready, so we didn't actually have any way to predict whether we're going to get binding affinity. And so what that means is we never explicitly knew beforehand whether our AI-generated biologics or antibodies would bind to the same target as Herceptin. So it was a significantly pleasant surprise that 69% of those 343 variants targeted the same thing as Herceptin. Now, it likely took years of painstaking laboratory experimentation to come up with these patented antibodies. Now, our algorithms did this in a week, and if this technology had been around 20 years ago, you can just imagine how it could have had an impact. So now I want to move towards the coronavirus itself. So one of our collaborators, which is actually a laboratory, wanted us to computationally generate a broadly neutralizing antibody against the coronavirus. Now, up until this point, before the coronavirus happened, our company was focused exclusively on oncology and autoimmune disorders. But as you can tell, it would be not very complicated to just change the input sequence, which would be something that can target coronavirus. So our collaborators wanted sequences that demonstrated affinity or binding affinity to the coronavirus, but they also wanted to ensure that it was developable, that you could make the drug, which is what our screeners do, and to produce these sequences faster than they could with their laboratory technologies. In this case, we took an existing antibody that neutralized the original coronavirus and applied our evolutionary models to it. Now, the reason we did that is because the SARS-1 and SARS-2 are very, very, very similar. They're mutated versions, right? Not to say that one is a mutated version of another, but they're mutants of one another. They're just mutational versions of coronaviruses. So in the span of a few hours, we generated a little less than 2 million variants of the SARS-1 antibody, which we then screened down using our in silico assays, our computational laboratory tests, to approximately 10,000 variants. We then clustered the remaining sequences, sent them to our collaborators, and actually checked the structural binding. And what we delivered to our collaborator is the most mutationally diverse AI-generated variants that have been pre-screened. We submitted those initial characteristics to our collaborators, including providing the mutational complexity, the technological complexity, and this is done for and then we work with them to actually choose the variants that they'll synthesize and express and test in lab. Now, this is done for 2 reasons. First, it allows us to, on the fly, provide an analysis of the variants, because we are providing them 1,000 antibodies, each with 200 data points. And so it's really hard for any person to wrap their head around 200,000 data points, so we can actually provide that analysis on the fly. Second is that it actually gives us a learning opportunity because, as I said earlier, we're not antibody engineers or laboratory scientists, so it's great to learn the diversity of thought from our partners so we can continue to make our algorithms better. And so we're actually expect those results in the next couple weeks, and we're looking forward to receiving them. So I want to tie this back to the business of why we're doing what we do. Currently, it takes almost 15 years to get a drug from a laboratory bench into the doctor's office or prescription. What's even more significant is the incredibly high failure rate common to drug discovery and development, which exceeds 90%. And that's the primary driver for the nearly $3 billion it costs to bring a drug to market, because it includes all of the failure rates and the huge amount of time. It's why in the United States, part of the reason why drugs are so expensive. And this is a serious problem that we're looking to fix. This is why our company Evolve exists, to use artificial intelligence to quickly design drugs that are less likely to fail, all at a considerably lower cost, so that we can have a positive effect on humanity, on society, and help heal, accelerate the speed at which healing reaches those in need. Thanks so much for listening. Oh, Jenko, I can't hear you. I still can't hear you. You're on mute. Jenko, you're on mute. Do I need to do something? I can't hear you. You're muted, Jenko. I see the mute button on for you. Yeah, just you need to unclick mute on the Teams app.
Okay, yeah, on the Teams app, yeah. Yeah, because there is a button here, there is a button there, too much of buttons, for this kind of online conference. So thank you a lot for a great presentation. You're quite precise, quite informative about what you were doing. We have one question. So there is also
apologize to anybody that I didn't answer in real time. I don't speak Croatian, so I had no idea what that was saying. I apologize.
Yeah, no problem. So there is a question. First of all, hi, great presentation. Could you further elaborate on the fitness function for the genetic algorithm you used?
Yeah, so the generative algorithm is basically it's a probabilistic model. So the challenge with the I can talk a little bit about it because we keep it a trade secret. So I apologize, I can't get too much into it. But the way that it works is that and let me bounce back to that to the actual image itself. So what's happening here is that the algorithm is looking at the I'll explain from a very high level. Takes this sequence, so any input sequence, and then pulls from a database of around 1 billion sequences right now to try using a hidden Markov model to profile hidden Markov model to find similar sequences, not across the whole space. So the best example that I've given is using the word mask, and we all know masks right now. If the algorithm's actually pulling from a very large database and it's trying to find similar patterns, so it might pull the word musk, or it might pull the word mast, or it might pull the word bask, but it won't pull the word plop, because there's no relationship between those two. So that gives us a sort of a what is mutationally possible. Then it looks at the probability of actually seeing a change in that particular position relative to all other positions. Now the challenge is you can't actually calculate that because it's too large. So we have to use stochastic gradient descent as a way to look at to estimate the what's going on in between the values. So this looks at the log likelihood of seeing that probabilistic change, given that given that the enormity of this, basically what you're seeing here is 120 by 20 matrix, which is basically 2,280 possible mutational changes. And so what the algorithm's trying to do is predict the log likelihood of seeing a particular type of change. And so hopefully that answers, Joseph, your question. And I'm happy to get more into it offline. What I'll do is I'll share my email address, and I'm happy to, and if anybody wants to email me, we can happily take this offline to discuss it a little bit further.
Yeah, of course. Why not? That's a great idea. Or they can write to the conference info mail, and we can share it if they like, you know.
For sure.
So there is another question.
Okay.
Thanks for the talk. A question from the biochemist here. Just to confirm, are you modifying anti-HER heavy chain in hope to find a new target for the app, or maybe to address the resistant cancer patient treated with MAbs developed? Why anti-HER?
Okay, so we actually just took HER2, an anti-HER drug, as an example. We can actually do this with any antibody or any actually biologic sequence. And let me just jump ahead because this is a really good question. So is it Tomislav that asked this question?
No. Biochemist asked.
Okay, so what I want to show you here is actually this box right here, that's 69%. What we're actually doing is modifying the antibody, but maintaining target specificity. So now we can create a more diverse set of, in this case, anti-HER2 drugs that may actually, in this case, it may potentially, because Herceptin actually causes cardiotoxicity in about 40% of all patients that receive it. And so what we're doing is we're creating modifications, not trying to hit other targets. This actually maintains target specificity. That's where the evolutionary model comes into play. It's mutating while maintaining target specificity. So I hope that answered your question, and feel free to ask a follow-up if it didn't.
Marina is from the University of Essex.
Hi, Marina.
So, okay, great. It's just around the time that we need to finish and wrap it up, because we have panel. It was really great to hear you. There was a few questions, so I believe there was a positive impact of your presentation to the guys, out there. It was quite precise, quite informative. I will share your email with everyone who is interested, so you can be in contact with the others. Sure. So thank you a lot for connecting. I hope that in the near future we can bring you here into Croatia.
I would love to.
If you've never been, I think that you will enjoy it. Unfortunately, now it's not the time to travel, but hopefully soon. And if your project becomes successful and you find a cure for a pandemic, then it.
Yeah, we're working actually on many different diseases. It just happens to be coronavirus is one of them. We're actually working on treatments for cancer, for other autoimmune disorders. Mental health, because our algorithms are sort of agnostic, and so we're actually very easy to switch. But very much excited to communicate more with the Croatian data science community as a whole, because we're data scientists, so it's always exciting to talk to more data scientists. Yeah,
okay, great. Thank you all once again. Have a great day. I hope there is still sunny outside in Miami, so you can maybe go to the beach. I hope you have a great success with the company, and maybe we can hear from you when you're going to be unicorn or something. So hopefully, great. Thanks again, and hopefully see you again.
Fantastic. I'm looking forward to it, and I like your hair.